Hello everyone. This video is about action potential propagation. So in order to understand it, you'll need to understand action potentials in general before you watch it. So if you're already familiar with this form of displaying the action potential, a change in membrane potential over time, or voltage, same thing, where initially there's a strong, fast movement of sodium into the cell and then potassium out of the cell, and a return to the baseline negative resting membrane potential, then you're ready to think about how these move. And I say move in quotes because the key thing for today is they don't move. An individual action potential is a local change in voltage. It can only occur, it is a thing that occurs in one place. That's what an action potential is. So if we're going to have action potentials at the axon hillock near the cell body, and then also down the axon all the way to the axon terminal, then we have to realize that that's not one action potential moving down the axon, but rather separate individual action potentials occurring sequentially from the axon hillock all the way down to those terminals. How is that possible? Well, if we think about what an action potential is, right, we know that it starts by some depolarization of the membrane, usually by an accumulation of positive charge from postsynaptic potentials, sometimes from stimulation at a sensory receptor. But either way, we get some positive charge, and that depolarizes the neuron to the point where the sodium voltage-gated channels open, right? And the sodium enters the cell. This is just a standard action potential. So the key thing that we're going to ask now, though, is what happens to that sodium? Because normally when we talk about an action potential, we just say, oh, the sodium comes in, the potassium leaves, end of story. But in order to get this action potential to move, that is, to create a new action potential, we have to think about what happens to that sodium when it comes into the cell. Well, lots of things could happen, right? It could move around in any direction. It doesn't know where it's supposed to go, so it's going to move randomly. And these individual ions, as they move randomly, many of them will run into the cell membrane. And at that point, they're likely to be caught by sodium-potassium pumps and kicked out of the cell. However, other sodium, by chance, will remain inside the cell, inside that cell membrane, and drift either upstream or in a retrograde direction, that is back toward the cell body, the left on our picture here, or drift downstream or in an anterograde direction, that is to the right on our picture here. Now, sodium that drifts to the left won't have any effect, and it'll eventually sort of spread out and dissipate, because to the left of our picture here, the sodium, any voltage-gated sodium channels that have already fired have been inactivated. So it's impossible to trigger an action potential there. We say that that period, that segment of the membrane for that period of time is in a refractory period. Can't fire another action potential. But the sodium that happens to drift downstream or in an anterograde direction is going to, by definition, carry positive charge to that next segment of the axon. It is positive charge, right? These ions are positively charged. But at the same time, the fact that the sodium came across the membrane into the cell means that the outside of the cell membrane there is relatively negatively charged compared to what it was before, and that will draw positive charge from downstream and upstream to that place, the place that's yellow in this segment of the picture, because this positive charge is moved into the cell, having become relatively negatively charged. A positive charge is going to be drawn into that area all along the outside of that cell membrane. The net effect of that, of sodium moving downstream and of some positive charge outside the cell moving upstream, is that the downstream or anterograde segment of that axon is going to be depolarized, right? Positive charge is moving there, and outside the cell, positive charge is leaving. So that segment of the neuron becomes depolarized. And if it becomes depolarized enough to open those voltage-gated sodium channels, then we're going to trigger an action potential in that segment of the axon. And once we trigger one there, this process can simply repeat. Sodium enters the cell. From that action potential, it drifts around randomly, but some of it will drift downstream and create enough positive charge to trigger the next segment of the axon to threshold. In fact, we can argue that the reason why the sodium electrochemical gradient is so large is in order for this to be possible, right? Or we can say it does make it possible to have this huge electrochemical gradient. And if it were possible to create this movement of sodium with a smaller electrochemical gradient, evolution would probably have settled on that method because pumping of sodium is very expensive. So we have a huge sodium gradient in order to get a huge, or that allows us to get a huge movement of sodium into the cell, which allows us to get a good enough movement of sodium downstream to trigger the next action potential. This process repeats down the entire axon. And it is a fast enough method to be quite effective in, for example, very small animals or in neurons that are communicating with other neurons that aren't that far away from each other, as often occurs in your brain. Quite effective. But if you have neurons that need to travel, that are covering a very long distance, such as neurons in your spinal cord whose axons go all the way down to your foot, that method is not fast enough. And so the method that evolution has happened upon in vertebrate animals is the use of myelin to allow this process to go more quickly. If you think about the problem with the method that we just discussed, the issue is that each action potential takes time. So it would be better if we could have fewer action potentials per unit distance of an axon, especially in a very long axon. We don't want to have tens of thousands of action potentials. It would just be too slow if you have to cover a long distance. So what a cell could do is it could only, right, an action potential can only happen where there's voltage-gated sodium channels. So if the cell puts voltage-gated sodium channels in areas that are spread out, not right next to each other, then we could hope, and at this point it's just a hope, that the sodium entering from one action potential would drift down the axon and trigger an action potential at the next place where there are voltage-gated sodium channels. However, by itself, this won't work because the sodium can't be expected to go completely straight down the axon because it doesn't know what it's supposed to be doing, right? Most of it's going to run into the cell membrane at some point in this long distance between areas with voltage-gated sodium channels. So, and again, if it runs into the membrane, it's probably going to get grabbed by a sodium-potassium pump and get kicked out of the axon. So the solution to this is to take these areas between the places that have voltage-gated sodium channels and insulate it. That is, to prevent movement of sodium out of the cell. In these areas, there's no membrane permeability to sodium. It can't move in, it can't move out, there's no voltage-gated sodium channels, and there's no sodium-potassium pumps. There's just this insulated area created by the fatty portion of these oligodendrocytes or Schwann cells, depending on whether you're in the CNS or the peripheral nervous system, that wrap around these axons and prevent ion movement across the membrane. In fact, they prevent the movement of pretty much anything across this segment of the axon. So when sodium enters from an action potential, again, some of it's going to move backward and have no effect because of the refractory period, but that which moves forward or toward the axon terminal or in an anterograde direction, all being to the right on our picture, is going to drift down the axon to the next node, that is the space, these spaces between myelin, these spaces with voltage-gated sodium channels, and trigger an action potential there. One way to think of it is like bumper bowling, that, you know, if you throw your bowling ball here and it's a bad throw and it would go into the gutter, well, it just hits the bumper if you have that bumper inflated in the gutter and comes back into the axon or into the bowling lane and is able to knock down some pins when you're bowling. Same thing here. Any sodium that would happen to run into the cell membrane here doesn't exactly bounce off, but it is unable to exit the cell, remains in the cytoplasm, and eventually creates enough positive charge at this next segment, this next node, to trigger an action potential. So in this axon, we wouldn't have action potentials right next to each other. We'd have one here at this node, and then one here in the next node. And the effect then is that it looks like the action potential is jumping down the axon. So sometimes this is referred to as saltatory conduction, or conduction of action potentials in a jumping way down the axon. Really, the action potential is not jumping because it's not the same action potential. It's a different action potential. But the benefit is you only have action potentials in a relatively few action potentials spread out over your long axon, so the speed can be much faster than that in an unmyelinated axon. Okay, I hope that made sense, and if not, feel free to ask me questions about it.